Okay, here we go. So this is a wrap-up of yesterday's discussion when we were talking about mitosis, meiosis, and chromosomal disorders and karyotypes and all that good stuff. So this should serve as a really helpful review. So, just to review, we have 46 chromosomes, and hopefully you can see that. 46 chromosomes in humans. That number changes from species to species. 23 homologous pairs to be exact. We do mitosis and cytokinesis to basically replace any cell in the body. But if we're trying to make sperm and egg, we go through a different process called meiosis. We do that so we can cut that chromosome number in half. Otherwise, if the sperm had 46 chromosomes and the egg had 46 chromosomes, you'd have new organisms that have 92. So we take those 46 homologous pairs and split them up to have 23 from mom, 23 from dad, combine them with a new organism, and now we have 46 new chromosome combinations, and it makes a lot of fun. So that's why we do that. In mitosis, you're trying to make replica cells. In meiosis, you're trying to make brand new combinations of organisms with chromosomes from within that species. So I think I've answered those questions right there. Any cell that's not a sperm or egg we call a somatic cell or a body cell. Anything that's a gamete or a sperm or an egg. So a sperm is the male gamete and an egg is the female gamete. They are haploid, meaning they have half the number of chromosomes. And that's easy to remember because haploid almost sounds like half. That is kind of shown by the number N, and most organisms are diploid, meaning they have two sets. So every cell in your body is diploid except for sperm and egg or gametes. We've already defined these terms right down here for the gene or an allele and all that stuff. Here's some other examples of diploid organisms and the number of chromosomes within those. So the karyotype is what we talked about. It's this looking thing right here. It just shows all the different chromosome pairs lined up, all 23 homologous pairs in the case of humans. And you'll see right here, this is important. 44 of those chromosomes are autosomes, and the last two are the sex-determining ones we call sex chromosomes. So here's all your autosomes, and the last pair right here, the X and the Y, are the sex chromosomes. So that's what we're talking about right down there. Females have XX, so all of their eggs have, when you go through meiosis, you split this into each egg. So half of the eggs have an X, and the other half also have an X. In males, half the sperm have X's and half the sperm have Y's. So females are XX, males are XY. So just keeping that in mind, it means that the male actually determines the gender of the baby, because no matter what, the female donates an X chromosome. I'm just going to let you read through that because I want to make sure that we get onto this next part here. So the whole process of meiosis, we did this with the Play-Doh in class. I know it seems weird if we're trying to take our 46 chromosomes and cut them into half. The first thing we do is double it with an S phase of DNA replication. So we take our DNA, duplicate it, we go from 46 to 92, and we have now homologous chromosomes. So that's kind of shown, they don't show up, but we have, it basically makes those sister chromatids. And the first thing they do in prophase 1 is those homologous pairs pair up. So it's really weird to think about. These tetrads right here, hey, come back here. They now have 4 sets of the same gene. Mom's and her copies from the S phase, dad's and his copy from the S phase. So anyway, this is one major difference from mitosis and meiosis. We have homologous pairs lining up, 1. Tetrads forming, basically the same thing as that first difference I pointed out. And another big difference is 2, they have crossing over. That's where they're going to exchange information between those homologous pairs. So that's what's going on here. 1 second. Sorry about that little interruption. Okay, so I think we just talked about these tetrads forming, so the crossing over could occur. Crossing over basically ensures that now the cells that are an end product cannot be identical. We've just literally shuffled the deck. So move on to, so just to review that real quick, there's an interphase, an S phase where we double the DNA. That's just like mitosis. But in mitosis, we do not have homologous pairs forming tetrads. That doesn't happen, and crossing over doesn't occur. So there's 2 differences right there in prophase. One major difference between metaphase 1 of meiosis and metaphase of mitosis is that now it's the tetrads that are lining up side by side, or the homologous pairs are lining up side by side, whereas in mitosis, the chromosomes line up single file. So side by side, and then single file in mitosis. So there's 2 differences here, 1 difference here. In anaphase, well, that also means that now you're pulling tetrads apart, or you're pulling homologous pairs apart, and you are not pulling sister chromatids apart. So basically, you need half the number of spindles in anaphase. And if you really think of this from a big holistic view, this doesn't really do much. All that really happens is homologous pairs come together, they line up, and they get pulled apart. So you're like, okay, hold on here. Why did they line up just so they get pulled apart 2 steps later? So the crossing over could occur. So the chromosomes line up, cross over, get pulled, or they pair up, line up, and split up. But it's this phase right here with the crossing over that's so important. Now, here's a good look of homologous pairs of chromosomes forming tetrads. Tetrads because there's 1, 2, 3, 4 little sister chromatids. And they switch the genes around. It doesn't make new genes, it makes new combinations of genes, okay? Then basically the rest of this is very similar to mitosis. This second phase of meiosis we call meiosis 2. So there's a prophase 2, metaphase 2, anaphase 2, cytokinesis 2. And there is no tetrads because they can't form, because the homologous pair is now in a different cell. So there's no tetrads, just like mitosis. There's no crossing over, just like mitosis. So that's the same, except that it's happening in 2 cells. Here's a good example of these chromosomes line up single file. So they're lining up single file in metaphase 2, just like mitosis. And when they split up in anaphase 2, they also are sister chromatids being pulled apart, just like in mitosis. The end process is 1, 2, 3, 4, unique, haploid, somatic, the end product is 1, 2, 3, 4, haploid, unique, gametes, whereas in mitosis, the end product was 2 identical diploid daughter cells. Okay, so now you throw little tails on those, you got sperm, and that's basically the end product. We've cut our number from 46 to 23. Okay, so let's real quick zoom in on that gamete production. So once again, 2N, N, and we split it down in half. This is not really shown well. Anyway, basically in males we produce 4 sperm. They're all unique. In females, when we make eggs, we take the cytoplasm out of 3 of these and move it over to 1 super egg. So technically, meiosis in males produces 4 sperm. Meiosis in girls only produces 1 egg, 1 ova. So that's one little subtle difference. The other 3 things are called polar bodies. Here's an example of some errors in meiosis. Now, every time I see this, I think about fixing it because it's a mistake, but that's exactly what this is all about, a non-disjunction. So what a non-disjunction is right here in the step of anaphase 2 and meiosis 2. These sister chromatids have been stuck together for a long time. It's very easy for them to break or not break, have the centriole spindle not break, and instead of separating these, it leaves you with this problem, where instead of having ooh, look at that. See, it should be 1, 2, 3, 4, 5, 6, 7, 8, and instead we have 1, 1, 1, 2, 3, 1, 2, 3. So that's not what we wanted. We wanted to have 2, 1 of each in each of these cells that we screwed up. So a non-disjunction is when sister chromatids fail to separate correctly during meiosis 2, more specifically anaphase 2. It causes the wrong number of chromosomes in gametes, either too many chromosomes or not enough. Which means that those, since that's the sperm and egg meeting, the organism that is the product of that afterwards is basically a clone of all those cells. I mean, even as a teacher, I don't think about this, but basically the first little thing you have is the reverse of mitosis. You have fertilization. Meiosis splits a cell down and makes a haploid. You have haploid and haploid coming together to make diploid and fertilization. And then after fertilization, you just have tons and tons of rounds of mitosis that turns that one brand new cell that was a half of a sperm and half of an egg coming together, that cell divides like crazy via mitosis to make a new organism. Well, if that sperm or that egg was missing chromosomes, every single cell in that organism's life and that new organism is going to also have that same screw up. So there's no fixing this. Every single cell is going to be missing or have extra chromosomes because of this non-disjunction. So just to review that, it goes meiosis to make gametes. Gametes come together via, during fertilization, and then it switches over to mitosis and just makes copies and copies of those identical cells over and over. So, okay, Mr. Archibald, we have screwed up numbers of chromosomes. Well, here's what it looks like. This is a karyotype. So now we're actually zooming in, and we have 23 pairs of homologous chromosomes. This top shelf stuff here, 1 through 12, they're huge chromosomes. You cannot have extra versions of these, or you're going to be an instant miscarriage. Chromosomes 13 through 22, they're smaller. So you can actually have extra chromosomes here, and it's not going to be good, but it may not lead in an instant spontaneous abortion. So you can see here, instead of having one pair of X's, we have one X, another X, and a third one. This is triple X syndrome, known as Electra syndrome. This makes a pretty normal female. She might have irregular cycles. She may have a strange, when she goes through puberty, it may be really early or really late, and her rhythms may not always be perfect, but for the most part, there are no major symptoms along with Electra syndrome. And if you look at this right here, we have 23 homologous pairs of chromosomes, an X and 2 Y chromosomes. So obviously, this will be a male, obviously, it's just they have an extra Y. We used to think that having an extra Y chromosome led to unusual amounts of aggressiveness, and we thought it may be the serial killer gene, but then we realized that no, this actually is a pretty common thing and doesn't really mean any side effects either. This, on the other hand, is 2 X's and a Y. Well, this will be Klinefelter syndrome. It's a male because there's a Y. The extra X chromosome creates feminine features when this young man goes through adolescence, and will give him some feminine features. This can often be not caught if the man is overweight, because a lot of these features can be attributed with being obese. This is going to be 1X. So instead of having an X and an X, or an X and a Y, it's an X all by itself. This is 45X, otherwise known as Turner syndrome. This is a girl who will never go through puberty and become a woman. Here's an example of 2 women whose age is not that different, but outward appearance looks very different because this young lady here has Turner syndrome and doesn't have the second X chromosome that becomes activated and basically turns her from a girl to a woman. She stays a girl her whole life. Linda Hunt, a well-known actress, is an example of a young woman who is dealing with Turner syndrome. It's the only monosome that doesn't result in immediate miscarriage. So in other words, it's the only time you can have a missing chromosome and not immediately be terminated. This you've all seen. This is trisomy 21, otherwise known as Down syndrome. In this case, it's a male. This is the video we watched today. This is Edwards syndrome, trisomy 18. It's pretty gory here at these parts. Most of these children do not live for more than a day or a few hours. This video, if you're really interested, you can watch, and it'll make you come face to face with Edwards syndrome. This is Patau syndrome. It's even worse. Don't do too much research on it. It's usually an immediate, it's basically a horribly deformed baby. And this is the trisomy 13 is as big as you can get, and every other chromosome here is too big to have extras of. That's Patau. Here's some good differences between mitosis and meiosis. I encourage you to look at this and study this. This looks a lot like a test question. Here's a great overview of mitosis and meiosis. Yes. Okay, that's basically everything I wanted to go over, so thank you very much for watching, and I will see you in class tomorrow.